I'm going to be talking about 1 million SARS-CoV-2 genomes. First of all, we know that viral transmission defines a tree. So you give the virus to somebody else, they give it to somebody else, and we have a transmission tree. Amazingly enough, with genomes, we're able to actually reconstruct this transmission tree. And what we see is in these colored ovals, mutations happen. On average, 1 or 2 mutations every transmission. And with 30,000 RNA bases, that means we get a unique signature for the transmission path as we trace out these genomes. This is the first time we have ever been able to, in real time, track the genomics of a virus as it spreads in a pandemic all around the world. And we can find things from this, as you've seen in the news, that are interesting. For example, we have combinations of mutations that are unique. And when we look at samples, say from Baltimore, we can say, wow, we must have had 2 separate introductions. For example, there's the orange blue version and the red green version. They came from 2 different places, and therefore we had 2 recent introductions into Baltimore rather than 1 introduction that spread to another individual. And this has helped us understand the dynamics of the disease. And when new variants have come out, it's helped us recognize that those variants actually pose a threat or not. And part of that is illustrated here. The blue mutation causes a lot more transmission than the red, and therefore you can actually quantitatively assess how dangerous new variants are in terms of increasing the rate at which the disease is spread. Now, of course, we have a constant battle that is going on between the host immune system and the viral capacity to mutate. This has actually been playing out for millions of years, and as we know, this plays out in the SARS-CoV-2 virus on the viral spike protein and the ACE2 receptor in the human genome. We can actually see on our browser, and I invite you to go to if you just say SARS-CoV-2 browser on Google, you'll find it. We have all the information over the million-year battle. The red is where there's been a lot of change in the actual human, in the virus in response, and on the right, change in the actual genome coding of the ACE2 gene on the right. And so you can see this play out. And you can actually map that to the 3D locations within the protein. Doing this, a number of researchers have now developed experimental systems where we can make speculatively changes in the spike protein and see what they're going to do. And this is some beautiful research on the bottom that's in our browser, along with 5 other studies like this. This is from the lab, Star et al. From a lab in Seattle. And they actually were able to say when and if the virus mutates in this way, that will actually be dangerous or not. So this is an incredible opportunity for us, the world, to anticipate the virus's next move. And again, massive genome sequencing, massive experimentation puts us in an extraordinary position for this virus and for new viruses if we can pull all this information together. So you can make specific predicted contacts, specific 3D analysis, and specific experimental analysis of the effects of these mutations on the virus. This is a funny thing. You know, we were talking about this months into this. We were saying, look, there hasn't been much change, nothing to worry about. And it wasn't more than 2 months after this that all these variants hit. And we realized that actually this was the name of the game. The virus is adapting. Here's some of the changes. You may have heard about N501Y. This is a specific mutation of an amino acid in the virus's spike protein. It actually occurred in 3 independent instances in England, in South Africa, and in Brazil. So we see that the virus is actually exploring the combinatorial space of possibilities in such an acute and intense way that it's actually finding this optimal change independently in different parts of the world. So we are up against a very systematic adversary here. These are the variants that you've learned about and that we're intensively tracking right now. We won't go into the details of those, but on our browser display, you can see how these viruses have led to changes in the dynamics. In particular, the gold at the top is the famous B.1.1.7. And you can see how it led to an extensive expansion of progeny. We now have more than 1 million genomes tracking daily, updated in this massive phylogenetic tree, all linked to the actual genomics and variants. This is an extraordinary tool. What we have are actually, and I want to talk about a little bit, end with the politics of this, there are actually two databases. One is the International Nucleotide Sequence Database Consortium, which has fully open data. They have about 500,000 genomes. And then there's the Global Initiative for Sharing Avian Influenza Data, GISAID, that has 1.3 million genomes. And I wanted to react to the first talk that we heard too, about hacking and data sharing.
David,
yeah,
we're done.
Go ahead. We're
Got our
at the
time.
end, right? Okay. So in terms of an open database, the INSDEC is open. You cannot hack an open database that everyone already has. So let's open up this data and let's get GISAID to open up all of these data. So thanks. You know, I won't go into the whole politics, but we received a cease and desist at one point from GISAID. We're fighting it. We have to share all of this data immediately. That's my main point. Thank you.